Okay. Just gonna do a little late night stream here. Working on setting things up a little bit, so bear with me. I've hooked my nice camera back up to the Light Microscope again, and I've been looking around in some samples that I collected. I went out on sometime over the weekend to a local park, to Deming Park, and took some photographs. I was out really just sort of playing around with my new lens on my very nice camera, and got some pictures of some woodpeckers mostly, and some squirrels. Just kind of screwing around with trying to figure out how the telephoto quality would be in bright light. We've had a lot of very gray days here recently, and so I thought I would sort of see how the lens quality was in full sunlight, and I also, while I was out there, collected a few sticks. And recently I've had a lot of good luck looking at lichen-coated sticks. The older the sticks looked, usually the better the sort of quality I could get with respect to finding water bears. So I went for a long time without being able to find any water bears, and once I sort of switched to looking at just sticks that have fallen on the ground, like old rotten sticks that have fallen off of trees, I pretty much can't lose with those. I haven't been able to, well, let's say I haven't had a collection fail me yet since I switched to doing that, and I've sort of got my technique down. Mostly what I do when I'm out collecting is just find any sort of stick that has a fair amount of lichen covering it. So for clarity, here's a stick like that I sampled for some of the material that we're looking at in the stream from tonight. And you can see that I've cleared off just a little bit of the stick between here and here. It's a sort of square shape as a result of that. And this part's still covered with the lichen. This sort of light-colored stuff is the lichen. And then all I did is I have a little paring knife. And I just sort of scrape the lichen with the paring knife into a little bowl. And so what I'm left with after that is usually just something that looks like, I don't know how well you can see that, but it's basically just a little stew of water mixed together with lichen scrapings. And this usually does a good job at producing water bears for me. So I haven't had any trouble since I sort of switched to this tactic for collection purposes. Hey, what's up, Mallory? And then I just I have a little pipette, and I've cut the tip of it off so that the lichen doesn't get stuck in the end. Yeah, that's what I'm doing, just chilling out late at night on my microscope. I spent the whole day grading, and I needed to do something that was, I don't know, either creative or different or whatever, but, got to a point where there's not really a whole lot I can do. It's cloudy out, and Sylvia's trapped our characters in Minecraft someplace where I can't leave them, and so I can't really screw around with that. And Sylvia and Carlin are upstairs trying to go to sleep. At least Sylvia is. So I put together some samples, and here we are, microscope time in the evening. And the samples that I put on here are from some sticks that I collected. And there's a water bear right there crawling around, a red one, or kind of a brownish red colored one. Right now it's on the other side of this pile of junk, the lichen that's there. It's not a kind of cool image. You can see through the skeleton of the lichen right here, and you can kind of see the algae that are visible through it. And the water bear is just kind of patrolling along the underside of it. He's a little camera shy, or he's just working his way around it. This one was on, he's got one of those, you can see on his nose up there, he's got one of those like star-shaped nose things. So sometimes they have those, sometimes they're not as obvious. This one's got a good set of them. We've yet to manage to catch one of these for the SEM that looks this nice. And this one's actually quite large. So we're looking at the microscope. Our settings are currently, this is a 10x objective with 10x eyepieces. So our magnification right now is at 100x. And that's the total magnification that you're seeing the image, and plus or minus a little bit. And then I could jump up if we wanted to. It's open sample. I don't have a cover slip on it, but I could get up to this, which is basically double the magnification. That's 200 times. There's a little bit of distortion because there's kind of an air bubble there, but I think you can see pretty clearly the water bear at that magnification. So it's just doing its thing, crawling around on its pile of junk, looking for food, scavenging for algae or whatever it comes across, probably. You can see that water bears have a really distinctive, all of them basically have this really distinctive shape, with 8 legs, with claws on the legs, and a little newt mouth, proboscis looking thing sticking off the front end. And the back 2 legs are actually a little bit different. They're separated from the front 6 legs, and I think they're mostly used for grasping. The other 6 legs are mostly used for climbing around on stuff. And yes, I cut my hair this morning. Got a little tired of it kind of like poking out from underneath everything. I still left most of my beard intact. I guess I just did a little cleanup on it. But, sometimes I just get a little too much for me to manage, so just clip it all off. See that the water bear is actually walking around in there's a piece of plastic in there with him. That bright blue stuff that's right here. Yeah, hi Alan. Hi Micah. Welcome in. Oh geez, let's not talk about A. That guy's so weird. There's a piece of plastic right there, like a plastic fiber. Bright blue color. Not sure what that's from. I scraped a lot of sticks before into this pile, so this is actually not a sample. That's just one of those sticks. And in fact, I think I scraped pieces from 4 or 5 different sticks in here, but I did a few sticks before I started and looked at just 1 or 2 samples from each of those and ended up with water bears in them. And I thought, oh, I'll just put them all together in one thing, they can all crawl around and hang out. So I've got a bit of a water bear farm going on as a result. A water bear is now trying to climb up the plastic. maybe it's decided that's not such a great plan. Here it goes. This is climbing around. Am I going to keep them alive through the winter? Well, the samples that I have, what I've been doing with...
Good news, everyone!
So we got a new follower there. Femme Fatale, welcome in. Oh, actually, it's Fetal Femme. The samples that I've collected from so far, because I can just get sticks whenever, what I've been doing is I keep them in a little bowl here for a while, take some samples out of it, and then after a while I just kind of chuck the whole thing out the door. Not the bowl, mind you, I do keep the bowl, but I just chuck a bunch of the water out the door. So incidentally growing a water bear farm in my yard as well, probably. I took them off of sticks and I just moved them into a new environment. I don't think it'll be a big deal because the ones that are in my yard also live on sticks. And, I throw the sticks out there too, so it's likely they could find their old stick again if they really wanted to. I figure they're just better off out in nature than trapped in my house at all. Although, I suppose I could keep some of them. Oops, what is this? Oh, that's a rotifer. He's just hanging out under that pile there. Not a water bear, but equally resistant. And there's a dead rotifer. You can actually see a lot of detail when they're not moving around. It becomes a little bit clearer what's going on with them. You can see, it's a little over bright in the screen. I don't know how to fix that really. Let's see if I can tighten down the... Oh, that didn't help. Oh, you can sort of see on the tail end of it right there. Two little prongs sticking off of the bottom. Those are actually its grasping feet. And then up at the head section of it, up here, you can kind of see its head and neck area. But because it's not moving, they're not nearly as exciting. When they're alive, they usually have their little spinny coronas out. And let's jump back out to 100x so I'm not scanning through material at a super high level. Here's another one. That's a dead rotifer, or maybe it's just sleeping. And I've looked through these samples a little bit. Primarily what I've found so far in them is the same things, the same usual suspects that we always see. Some water bears, some nematode worms, and some rotifers. And occasionally I find a dead mite. Although yesterday when I was looking through the samples, there's something in there crawling around. Is that another water bear? I might have to leave it here for a second and see if we can get him to come out. It's either a rotifer or a water bear. I saw little legs, but I didn't see whether it was... Oh, here he comes. I think maybe it's a rotifer. They tend to just be attached in one point and then sort of jump around from there. And I saw a bit of its body without any legs sticking out. Hey, Pantene, how's it going? And then I saw a mite the other night when I was looking around that was actually alive. I think it's still on one of these slides around here. But it wasn't moving very much, so I don't think it was long for the world. But that's pretty much what you find in the the lichen every time. I haven't seen a whole lot of other random things in there, and that's largely because you have to find organisms that are basically adapted to these dry conditions most of the time and just get wet a little bit now and then. And then they basically recover from that. There's something in there, kind of crawling around. Oh, that's a rotifer in the foreground, and maybe another rotifer in the background. And this is a wiggly nematode right here. Hello, little wiggly nematode worm. Still a bit over bright. Maybe I can fix that on the camera here. Let's see. I want to mess with maybe the ISO. That was the wrong button. No. Not having much luck making adjustments to that. Guess I could just turn down the brightness here a little bit. My camera kind of just makes adjustments on its own, so, you know, adds to the complexity of getting the image perfect. And my camera to be a little stupider, then I would have no problem with it. It's just a little too smart for itself. Here's a rotifer that's living and it's not hiding under a bunch of stuff. There's two of them actually right there in the field of view. This is a typical Bedeloid Rotifer, kind we might normally find in these environments. You can see it's attached by its little foot with a claw, and then it's got an antenna sticking up, and its head, and at the end of its head, just little cilia that spin around and through the water, draw things towards its mouth. That's where they get their name, rotifer. There's another one up here, inching along. They just have that one leg with the clawed sort of tail on it. Here's another one. This one's got its corona out, and it's a different variety or different species. A little bit bigger, and also it has these the really big corona sticking out the top. When they pull it out here, we'll be able to see it hopefully. So that is my camera. It's not a new camera. It's actually an old camera. Well, I mean old. I've had it for maybe a year and a half or something. A year. But I did get a new lens, and I use it on this camera. So, but it's the camera that I primarily have my lenses mounted on normally, rather than mounted onto my microscope. So the microscope mount that I put on here allows me to attach my camera to the microscope directly. And as you can tell by this sort of like ring around the outside edge where you can actually see the round shape, it's not perfectly fit to the microscope. That's because I have a 0.67 multiplier in between right here, and I probably need a 1 times magnifier in there. Hey Sarah, how's it going? Welcome in. I'm just looking at some lichen material that I collected from Deming Park, and I scraped off some of it from sticks, and found a bunch of rotifers so far, and a little nematode squiggling around, and had a water bear on here a little bit ago. I've moved away from it. I'm sure it's still in here somewhere. It's got nowhere it can go. That is another Bedeloid Rotifer. You can see this one's got sort of striations in the midsection right there. Right down the middle of its body, those sort of long muscles or whatever. that's a multicellular organism, and as I mentioned, it's got little cilia up around its mouth, these little things called corona, and it sort of spins them around a bit. It doesn't actually spin them in a circle, but it looks like they're moving in a circle. And then that produces a small current that pulls things into their mouths, or towards their mouths. I saw some on Pacific Plankton stream the other night, and it was hilarious because it had like a long Oligocyra or something, Melicyra, that was right next to it, and it kept spinning around and hitting it in the face every time, like while I was trying to pull stuff in. Hey, look! What's up, Mike? How you doing? And also Lex. Welcome in. Just looking around at some stuff in the microscope for funsies. I needed a break from grading, and I've had kind of a busy week. Monday I was just trying to recover from just, like the holidays. Well, we didn't go anywhere. I've just been so exhausted from this whole semester that I just kind of hung out at home and did nothing for most of the day. That's maybe an insect carapace. For almost the entire break, I couldn't really function. I just needed to recover. It's been a rough semester. And, oh, wait. There's a little mite right there, and it's living. There's a mite. Yeah? I think it's some sort of little water mite. Or an insect, actually. Because I think that thing only has 6 legs. Yeah, I think that's some kind of an insect. I'll zoom in and take a look at this weird little insect. Might be a mite. Yeah. Oh, hi, little chook. You're still buried in grading. Anyways, so Monday I was just like, well, I gotta get back into gear, and I spent like 2 hours just trying to figure out like what was on my calendar for this week, because it's just been brutal. And then I've just been in like grading and dealing with grad student committees that I'm on, just like one after another, while I'm trying to do other stuff around it. So thanks for hanging out, little chook. You don't need to, do any more than that, of course. Good. If I actually wrote things to people on paper and gave them out to them, it might be a little less taxing, but instead I have to read through stuff and type up like how to fix what they wrote. And if you just do it for one person, it's sort of okay, but like when you have to do it for 10 people and you have to do it like 6 times, no, it's a lot more work. And, I'm trying to help them learn how to write better, so I don't just want to go, here's a letter. I actually have to kind of skim back through it and tell them how to restructure things or whatever else. So, yeah, term papers, that's what it all gets to be about. Yeah, it's definitely got 6 legs, so. What's going on? Am I near? Oh, I'm on the edge of something really big here, and it's got a little bit of an air bubble on it. Let's see if we can zoom in on that. Oh yeah, I didn't want to bump that big block of junk that's next to it. So, my microscope is actually geared for looking at things. It's a high-powered microscope, and when you get in close like this and you start getting in the 200, 400 time size range or magnification range, stuff looks great. I mean, that looks really great. You can see all kinds of detail. Look at those like weird wrinkles in its chest area in there. And you can see its mouth. You can see those wrinkles right there on the side of its body. But you don't get to see the whole organism very much. So that creates an issue, of course. You gotta like focus in and out to actually see what you want to see. And of course, I didn't put a cover slip on this, and my objective is bumped up against that big black blob, whatever it was over there, piece of lichen. So whenever I move my camera, it also squishes the water around. But yeah, this guy's he's pretty active. Usually these things, I just see they're like corpses laying around. I'm not sure what it is. There's a good view of its face, or its head anyway. It does have little like tube-shaped sucking mouth parts like a typical insect. Doesn't look like it has any eyes, but sometimes those are really small on insects like this. Looks like it's found a little bit of food. We can also do this to zoom in. So if I wanted to get really close, the camera actually lets me get pretty close. Even closer if I wanted to, but I want to kind of like hit some sort of middle ground in here where we could actually see stuff. That looks like it's pretty good. Get a really good sense of what that thing looks like right there. Yeah, it's got a plate on its head, and then it's got those weird ridges all around its body. See them? Just kind of like hanging out, crawling, looking for food, doing its insect things. Its little spine sticking off of its body everywhere. I don't know what it is, but I've been kind of fixated on things that aren't water bears. I guess oh, you can see it's got a little antenna. See the antenna? It's got a little antenna sticking up off of its head. Like a little insect. They have sort of paddle-shaped antennas. And then those weird, crinkly sides on its body. So one thing that's cool about this, of course, is I don't know, these bugs probably like to be in the water. But, you know, it was living in, or hanging out in this lichen before the water got there. Just doing its normal insect things. It may have been hibernating the same way that the rotifers and the water bear and the nematode worms do. When there's another, what is that? Stick your face out. I'm pretty sure that's a rotifer. Let's see. Let's take it off of this crazy zoom I'm on. We can look around for some more things. There's a dead rotifer. You can see they have this like structure in their neck area here. That dark red spot. If you kind of look at it, if I can get the focus just right, you'll see that it's actually like a little plate. And when they're living, that thing, I think it's called a mastax, they can kind of move it around and chew stuff. So they pull stuff into their sort of head, throat area, and then I think that's the passageway that goes to their stomach. So keep an eye out for it when we see some living ones, and you probably will see it moving around in there. It's like a darker thing in their bodies. Also, occasionally when I'm rolling around in these samples, there's another insect. That one's dead. I'll come across water bears that are not active. Either they're still curled up in their little ball, called their ton state, or they're unrolled, but they're just not moving at all. Like they're just kind of sitting there. And
good news, everyone!
Scared the crap out of me. And when I find them like that, I think, oh, I found a dead one, and I'll try to transfer them over to cover slips so that we can maybe look at them on the SEM, because I don't want to kill anything if I can help it. And putting it in the, there's another insect right there, that one's dead. Putting in the SEM will definitely kill it. No matter what anybody tells you about water bears, they can't handle that gold plating plus vacuum thing, and they're not going to live through it. But so I find one and I transfer it onto like a cover slip, and I think, oh, I've got this thing, and it's all laid out. It's gonna. Is that a water bear? It's a rotifer. Sitting right in the bubble. Pretty sure it's a rotifer. Anyway, I think, oh, that thing's dead for sure. And I put it on the cover slip, and then I just set it there and let it dry, thinking it's going to dry out. It'll stay in its shape because it's dead. And sure enough, when I come back later, it's rolled up into a little ball. So I think they just kind of go inactive sometimes. They're not actually dead. I don't know if they're sleeping. I was going to Google earlier, do water bears sleep? Because they do just sort of, sit there sometimes. And I sometimes come across them where they're inactive, or I think they're dead, and I stare at them for long enough, and then their little claws move around, like just the claws, just like a little wiggle, like I'm not quite dead. And it's really funny to me. I don't know if they're struggling at that point or what's going on. So I was playing around the other day also, and I did this weird thing, which is like putting my camera not quite between the filters, and I get this really dark field kind of appearance. Let's see, what's it say? Sarah says, tiny antenna. I don't know if they sleep. I'm not sure what level of brain activity is needed also. Yeah, I don't either. Something I needed to just look up, I guess, and see if anybody knows. But I don't think that they can curl up if they're, into their ton state if they're dead. I think that we've come across some that were dead and that stayed that way. Like they didn't move for days, and they dried out, and they stayed unrolled up. And so I have some over here that are transferred onto cover slips that are in their ton states. They're rolled up, but you can't really see them anymore. I mean, they just look like a little block at that point. And those things that they roll up into are pretty secure. They're like a tight little spiral with, sometimes you can see a couple of the claws sticking out, but like that's it. So it doesn't look so hot in the SEM. I mean, you can see something, but it's not as exciting. So I thought, well, sometimes it's a miss, sometimes you get them. But at least in the light microscope, I can keep them, you can see them crawling around, living, so that's nice. You can find where you left them. I swear I left my water bear around here somewhere. There's that insect carapace I keep coming across. I probably got into one of these pockets in here, and then, you know, there's the water bug. The other thing is, if I get, you know, we exhaust all of our looking around options for it, I can always just put some more material in, look at a different sample. One of the great things about taking samples from sticks is that there's sticks everywhere, and the lichen on the sticks everywhere. So it's not like it's hard to find, and it's not like I can't just make some more, like look at another slide. So, oops, there's a rotifer. Right up at the top edge. There's another little rotifer crawling around, one of those Bedeloid Rotifers. There's another one. There's a nematode. There's a big rotifer right there. You see his big foot? He has 3 little claws on his big foot. Just on the one end. I want him to come out here and open up his corona and start spinning it around, because I'm sure he's one of those that has the huge corona. In one of our streams we saw water bear carapace with little eggs in it that were crawling around. I don't know how the eggs were moving, but they were moving around inside the carapace a bit. And some water bears will do that. The moms, they're mostly female, I guess, will shed their... There's another insect right there, there's a big one. It's a sort of similar to the one that we saw, or maybe it's the same kind, but that one's dead. They'll shed their skin, and then they'll just leave their eggs in the skin. So, as a way to protect their offspring. We're back to that thing.
You streaming?
Yep. Oh, there's another little thing. What is this? Now that's a mite, I think. That one has 8 legs. That is for sure a mite. I wouldn't confuse it with a water bear. It's got a segmented body. It's got little hair sticking off of its butt. That's definitely a mite. Let's see. I will jump over to the 20x. Let's see if we can see a little piggy. Do you see his little feet? Little piggy feet. There's his head. And then on its tail it has 2 little hairs sticking off of its butt. What's this? There's its face, right there. A nice close-up of its face. Definitely a mite. 1, 2, 3, 4. So on one side of its body it has 4 legs. They're not like the water bear legs. They're not like a cluster of 3 and then 2 separate legs at the back. They're just sort of evenly spaced down the body. And...
Good news, everyone!
Oh, we got a new follow. It's Barbarian Hamster. Oh, I only type copy pastas. Mites are small arthropods belonging to the class Arachnidia and subclass Acari. Mites are not a clade, as they span 2 different groups of arachnids. The acariforms are sister to the camel spiders, while the parasitiforms are sister to the false scorpions. Also, they exclude the ticks, the order Oxidida, and although ticks and mites are closely related, mites are distant related to spiders and scorpions. That's a wall of information. Thank you. I am definitely not an expert in mites, or really, for that matter, insects or arachnids. I mostly look at diatoms, but sometimes you'll find diatoms in these environments, growing on trees or in bark. There's very specific kinds of diatoms that live in those. There's also some, I should point out, there's our little piggy mite. Oh. There's a nematode whirling its way around. And down here, a rotifer. One of the big fat kind, juicy type. You can see its little antenna sticking up. I see something also back here in the background, crawling around underneath this pile of junk. Could be anything. It's probably a rotifer. There's a lot of rotifers in the sample. There's a dead Bedeloid Rotifer. There's a rotifer, another Bedeloid. I mostly study diatoms, as the name implies, but occasionally I like to look at other things that are also in the same sort of microscopic scale. And mites are a challenging group, as I'm sure Pasta knows. And that's because there are a lot of different types of mites, like an enormous number of them, thousands of species of mites, and or maybe hundreds of thousands is more accurate. And I don't think there's a lot of people who study them, so there's a lot of unknowns. What is this? Another rotifer. So we see them quite a bit looking at soils and looking around in, especially where I'm looking here in these sorts of things. We find mites and those weird six-legged insect things that we just saw. I'm not sure what those are. Also see the skeletons of Arcella, the sort of amoeba that sometimes live in these environments. I saw one of them earlier. I think my sample is starting to dry out a bit, making it kind of challenging for many of these organisms. It might be beneficial for the water bear if the sample were a little bit drier. I might be able to walk around on the glass a bit easier. You can see this sort of bubble around everything. That's actually the thing is wet and everything around it is kind of starting to dry up. There's a little nematode struggling with it. I'm going to jump over to another sample. That one's completely dried out. I was looking at this one a little bit earlier. Also saw a water bear here somewhere. Rotifer. Pieces of rotifer. Oh, there's an Arcella skeleton right there. Arcella. One of those little Mastax pieces. It's been a while since I did a stream from my microscope from home. A combination of holidays and some family stress, not associated with the holidays. And so I just haven't had it in me to spend time on the microscope, or, as I mentioned, I just wasn't doing anything for a while at the beginning of break, and then I just kind of stayed that way. I just need to basically recover. So I didn't even do any like regular streams for a week or something. Oops. There's a nematode squiggling about. And I was doing a lot of streams with my daughter, she was playing Minecraft while I was trying to figure out what was going on. And those are pretty funny. And I like doing those because I don't have to do much work. She does most of the entertaining, and I just ask questions. But she's convinced me to play Minecraft with her now. So, now I sort of understand some things about Minecraft. There's still a bunch of stuff that doesn't make any sense to me. But she has more fun just playing with me, I think, than streaming it at the moment. So we've been doing that. She hasn't asked me to stream it, so we haven't. There's a rotifer with its little corona going. It's one of those big ones. Let's see. So we've had some really nice sort of Minecraft time with our community. Hanging out in Discord with them and just kind of playing with my daughter as the community all together. That's been nice. And I think she'd rather do that than sit around building things for people, but it'd be fun if we went back and did a few of those streams again too. So, a lot of the same stuff, of course. Maybe I'll make a fresh sample. Time for you to head out. Okay, thank you. Auto mod. Held a message for reason identity. Allow will post to chat. Oh yeah. I'll allow it. Here you go. Sorry, Jack. I think that six-legged bug was a larval stage of a harvest mind, a chigger. Oh, okay. A lot of images of them show them appearing having 6 legs. Okay. That's interesting. They start off with 6 legs and then they grow 2 extra legs. That's super cool. Also, super confusing. But I didn't do any research on them, so I'm just going to trust what you think it is. That sounds really neat. And also, thanks for looking that up. The other 2 are small. Oh, I see. It had 2 little legs. We just couldn't see them. Maybe. Sometimes they're tricky like that. Where they... the legs are hidden from what we can see. Or they just look like antenna or something else, you know. Sometimes hard to tell. It had sort of a mite-shaped body, but there's another rotifer. It's not moving. It's probably dead. There's another arse.
Good news, everyone!
Those things always scare me. It's Blue Spider Monkey. Welcome in. Search Google for Well, I can't even say that. Okay, I'll look it up maybe and see if I can find it. I could also go back and take pictures. I mean, I do have my camera hooked up to it. I'm just, you know, I guess it's not that important to me. Here's a little water bear. It's like the ones I was talking about where they don't move. It's just sitting there. Like not really doing anything. And that's the right shape for a water bear. See how it has 3 legs in a bundle and then 1 set of legs back here in the tail end. And that's its head up here. The nice part about it not moving is, of course, allows us to focus on some things a little bit better. We can zoom in on it. Like real close if we wanted to. There's a nice view of it. You can see the mouth right there. It's the little newt face it has. A little, you know, proboscis-like nose. And if I focus just right, you can see the claws on the ends of its feet. So water bear will always have little claws on the ends of their feet. And they're in bundles. I think it's bundles of 2 or 4. And each of those has a series of smaller claws, and it looks like this one had a meal. So maybe it's sleeping, or maybe it's like, you know, like some of us have had a Thanksgiving dinner. And it's just kind of laid back in torpor, resting. Could also be dead. It's always an option. Just, in my experience, sometimes they just start moving again. So I don't trust that they're dead anymore. They could just be tired. You know? They've had a hard day crawling around in the lichen, and they just don't care anymore. I'm just gonna lay here. That is a rotifer. There's another rotifer up there that's dead, or inactive at least. Here's another one. There's a lot of rotifers in our current sample. See if we can find a water bear that's actually active. That's a rotifer. You can see that little mastax right there in the center of its sort of neck area. And then you can actually see the intestines, stuff in it. The dark sort of blobs down there around the bottom, I think is its little intestines. Sleeping its meals off. Hey Dozy, welcome in. And also Mama Bonbon, it's nice to see you guys. Very nice as always. Yeah, I could just be sleeping a meal off. It's possible. We'll wish him the best. Let's not disturb his rest. We'll just move on. You know, there's always another water bear somewhere in here. Or another pile of junk to look through. Maybe we'll find one crawling around. I didn't do a great job breaking that clump up. There's a big clump of darkness in there. Too much mass. Sometimes when I first collect these samples, the water bears are still in their tun state. And I think, I could just leave the camera on this thing and it will slowly expand out to its full size, like somebody's inflating a mattress, you know, like they just start to pump up and then they start wiggling around. And I've done that before, but I couldn't get the stream going before, you know, I wasn't prepared for it. I was looking around in the sample and I found one, and then it started expanding up and I was like, oh God, I can't get the stream started before this thing actually, like, is moving. And by the time I would get the stream going, it would already be up and moving around. But I thought it would be cool to have that where I, like, start one off in its tun state, and then we can watch it like come out of it, wake up. I just need to prepare a little bit differently. And I have some water bears that we have named. So there's the first water bear that Sylvia found on her own over there on a slide that we named Gubu. Gubu still lives, as far as I know. He's stuck over there on a slide, dried to it. And also Tardy B, the very famous Tardy B, is still on a slide over here with me. And as far as I know, she's still alive. So we could always put drops of water on that and try to find them and catch them coming out of their tun states, because they definitely are dried up over there. But I just didn't want to wake them up yet. Maybe we'll do that special stream where we wake Tardy B up. There's one of those little mites again, whatever they were, chigger mites. But the Jack thought they had ID'd for us. It's hiding under a pile of junk, but I recognized the shape. Also, I saw one of those little paddle-shaped antennae that it has. Not sure what this thing is. Looks like a little egg or a cyst of something. I get the I've had this habit of having people help me by reading the chat for me, and then I forget. Oh yeah, occasionally I need to actually read the chat. So if I miss something, I'll get back to it. This is a nice picture right here. So up here, one of these little tick bugs. That one has 8 legs. So I can see 4 on one side of its body. So I'm pretty sure that's a mite. So in our field of view right there, one mite. I can just do this so you can all see it with me. There's that guy. You can read the chat for me. Thanks, OpenSet. Welcome in, OpenSet. I can do this. I'm not totally incapable of managing my own stream. I can do this. I can type squad. There you go. There's some people you could check out that do microscope streams. OpenSet is one of them. Haven't seen OpenSet do a stream recently, but I hear he's going to start having some kind of a schedule, so we could know when to expect him, instead of his normal Spanish Inquisition sort of approach to streaming. Anyway, down here, a nematode worm, not far from where we were. Up here is our little mite. Down here, our nematode, and then right next to it, a tiny water bear. Look at him. That one's crawling around. The little baby. I don't know that it's an actual baby, just as a disclaimer. You got back started last night. Oh, okay. Oh, you're just going to be streaming a lot. Well, that's good to know. Let's see, Dozy asked, before I skipped over them, I didn't mean to skip over you, Dozy. Where is this sample from? These were collected from some sticks. So let's see if I can get all these little... Can I get them all in the same field of view? I think I can. Yeah, there's the whole group of them. Up at the top is our mite. In the middle, a water bear. On the left, a little nematode worm. I collected some sticks from Deming Park, which is just a little local village park here in Terre Haute, and hopefully you can see that. So that's just some lichen here, and down here the light colored stuff, and the dark part in here is just bark. So what I've done is I took a little paring knife, this one in fact, and I just scraped it like this, and I cleared off a bunch of the stick, and And when I did that, I cleared it off into a little glass jar. So, and I did this with about 5 different sticks. This is one of them. So those water bears and other things were living in this sort of crusty material here. And the other sticks I threw back out into the yard after I cleaned them off in part. And then what I did was, you can see there's a little stew here that I made by taking that, taking the see that green stuff in there is the lichen, and my pipette sort of sticking into it. And I just mixed it together with some real pure water. So in my house we have reverse osmosis purified water because, I don't know, my wife's father bought us a RoPure system for drinking water. So it doesn't have any of the chlorine or other things in it that our local water would have. And then I just made a little bit of a stew out of it, basically, with the water. So they have a little park for them to live in. And then I took a little plastic pipette. I have a bunch of pipettes because I'm a scientist. And I cut the tip of it off so that the lichen wouldn't get stuck in the end of it. So it's got a fatter tip than they usually would have. And I just took that and stuck it on a cover slip. Actually, these are on the whole slide. So, stew not for eating. I mean, you could eat it if you wanted to. I don't recommend it. Just lichen. I don't know. There's nothing in there that probably would kill you. It just wouldn't taste very good. Let's see. So Dozy said, crazy to think all these little guys live on there. Yeah, and in fact, all these guys were basically dried up on the sticks. I plucked these out from under a tree in the park where they'd fallen on the ground. And I mentioned earlier in the stream, I was actually out taking photographs. I wasn't really out hunting for water bears, but I thought, well, I'm in the park, and I should see if they've got any different kinds of water bears than the kinds I have at home. And these are basically the same kind that we see. I'm not an expert, but this little clear one, we see something like that here. And then there was like a red colored one with a sort of like a pronged snout. I've seen that one here in my driveway as well. Sticks that I collected off the roof of my house as well. So you can see he's got a little full belly, but he's still active. I should say she, probably. And nematodes were all dried up. Everything was all dried. So it's not just that they're living in those environments, it's that they're able to come back to life when you just add some water, which is kind of cool. The nematode's not doing so hot. But we saw some other ones earlier that were squiggling around quite a bit. This is a little tiny water bear. The red one that we saw at the beginning of the stream was probably 5 times as big as this one. This is just a little baby. Let's see. I'm going to make him bigger for you so you're not looking at the glare of my camera. And so you can see him. You see him crawling around. Water bears are sort of made famous by the fact that they're resistant to all kinds of things. Nasty chemicals, vacuum of space, super high pressure, very high temperatures, very low temperatures. In fact, there's many water bears that are sort of specialists for colder temperatures. I should point out, though, that they're not really, these aren't really environments that they prefer. Some of them do. Some of them prefer cold conditions, and some of them will only come basically back to life when the temperatures are really cold. Oh, the square grid's just it's an artifact of my camera. So I'm using this, my actual camera, and they have a conversion for it to make it into a webcam. So I'm running that software, and it's not used to being super zoomed in because it's supposed to just be like a webcam. So whenever I do that, basically you get the little square artifacts. Those are just rendering issues that it's having with the camera. So But I like this a lot better than the camera that came with my microscope. It's a lot higher power, and you don't get the weird distortions when I move. Yeah, it's a pretty nice looking view, especially when I zoom in, and you can really see a lot of the detail. You know, this is the 2 lowest powered lenses I have on my objectives I have on my microscope, and one of them doesn't actually belong on this microscope. It's just staying on here because it's what I have. I can turn the light down just a little bit and increase the relief. Right there, you can see what it's been eating. So, oh, it's cyanide teacups. Hello, cyanide. How are you doing? I haven't seen you in a stream for a while. You sort of lurk, but you don't talk. Yes. So, can it solve puzzles? I doubt it. I don't think they're very bright. You know, look at it. It's it can't even figure out where it's supposed to go. It's like a little helpless baby, right? I think the only puzzle it can figure out is, am I near food or not? And that's about it. They're not the world's smartest creatures. You can see its mouth though, and its throat when I zoom in like that. You can see the little food tube. See the food tube in its head? Look at that. So it's got the little newt mouth in the front, and like a little vacuum, they suck out nutrients and moss, lichen, bits of algae into their mouth, and then you can actually see it sitting in its stomach. So that green blob down there is just whatever it's been eating, probably a little bit of algae out of the lichen in its stomach. And this one's transparent. But you can see the little claws on the end of each one of its legs as it's sort of swirling around. And also, if you catch really carefully, you can see the end right there. It's got, the tail's sort of like shrimp-like or something. It's actually 2 legs that are sort of close together with claws on them, and they're really just used for grasping things. They don't have the like crawling ability of the other legs, so you won't see them kicking those back 2 legs very frequently. They're just kind of sit there unless they're grasped onto something, and it helps them get around. So, all right, Dozy, thank you for hanging out with us. Hopefully we'll see you on Saturday. We're doing a little bit of the Artist Back Alley Raid Train, and I should give a little shout out to Dozy. Sorry, it's really hard for me to type because everything's in front of my keyboard. Dozy Streams. I think I got that right. And you should check out Dozy. I think they do some tattoo art and stuff like that. Let's see. Cyanide Teacup's also a streamer, and I will give Cyanide a shout out because she deserves it. And we raid Cyanide regularly. She does a lot of great streaming of video games, and does a little bit of pixel art, and she's just got a great personality. You should check her out. Here's that little guy. Let's have to zoomed in on the 10x. Let's see. Lurking is your main occupation. Has anyone done a test? Looks can be deceiving. I don't know if anybody's tried to test the intelligence of water bears. They've done a bunch of tests on what they could stand by putting their tons states, putting them into their ton states, and then putting them through all kinds of crazy environmental damage, like UV radiation, and also non-UV radiation, like the really dangerous kind. And I think that they put them in temperatures as high as 300 degrees. And I think that's 300 degrees Fahrenheit, but I'm not positive about that. And they can live in that state. In that ton state, they're just like hibernating, basically. It's a cryptobiotic state. They're not moving. They're like curled up in a little ball. but they'll put a bunch of them in those little balls in these conditions and then basically put them back into water, and they test to see whether they can kind of come back to life from it and whether they can also reproduce on the other side of it. So those are characteristics that they test. I don't know how you would test the intelligence of the water bear or whether it can solve puzzles. I mean, bees can solve puzzles. But you know, I guess if it's the right kind of puzzle, I don't know how you set up something for organisms this small to test that. Let's see, where are we? Top, middle, slightly to the right. See if I come back to this guy later. We'll look around for some more. Oh, there's another one of these little mites. If you're here for the water bear and we keep seeing mites, I don't know what to tell you. They're in the lichen. Also, if you're here for the water bear, and we sometimes will see rotifers and other things. So, there's a dead rotifer right there. You can see the tail really well. A little two-pronged tail. Sometimes they're three-pronged tails. I mean, maybe it's sleeping, but I think that one's dead. See this guy? He's all covered with those little hairs. It's another one of those little mighty bugs. I see those carapaces all the time. I don't know if that's... That one looks dead. It's a rotifer. Oh, I should also zoom back out so you guys aren't like completely lost. A lot of the same stuff. There's another rotifer, a dead one. I want to see one of these big juicy ones open up its corona, spin them around for us. Oh, what's this? What did we find? Oh, it's a rotifer, and a little rotifer, and a big rotifer. There's one of the big ones. And that right there looks like a dead rotifer, or it's still in its cysted state, maybe? It's a little rotifer. We're in Rotifer Village right here. Bunch of them. There's another one, a dead one. There is a ciliate. We did find something else. There's a little ciliate spinning around. You can see that guy, right? If I were to leave this sample in the water, like in a wet stage for a week or something, and come back, it'd probably be mostly ciliates. They can reproduce like crazy, and they live on things that are decaying. So probably a lot of the things in our sample are protected by the fact that they get dried out, and the ciliates don't have time to like take over the whole environment and cause everything to decay. Dead rotifer. Sometimes I see a bit of the stuff moving. That's a rotifer. Another little spiny looking rotifer. Those Bedeloid Rotifers, I should point out if you weren't here when I mentioned it at the beginning of the stream, they are just as hardy as water bears, and actually in some ways more impressive. The Bedeloid Rotifers, as a group, have lost sexual reproduction entirely. They don't sexually reproduce at all. And usually for organisms, when they switch to entirely asexual reproduction, that's the end of the line. They usually die off somewhere after that point, because eventually the organisms get some sort of genetic deformity, and when that happens, the organisms can't live anymore, right? They can't reproduce or something like that, or they get a deformity that basically gets handed down, you know, and in a matter of 1,000 or 2,000 or 5,000 years, basically there's no more of those organisms living anymore. And so that usually results in them completely dying off. Like, there's almost no examples outside of the Bedeloid Rotifers of organisms that have switched to asexual reproduction and do nothing except for asexual reproduction, and yet, at least in the animals, have continued to live. Oh, here's a big a big water bear. So there's another one. We saw the little guy that was clear. This is one of the big ones, and he's a little kind of reddish brown. I think he's one of those armored types. She, I should say. Probably. Yep. And really active. No sex. Let's see. Do they have neural networks? I mean, I think they can feel things, so I'm going to say yes, and OpenSet agrees with me. They have a basic nervous system. Now I want to read all about their nervous systems. Well, that's, I think that would be a useful thing to do. You should do that. Let's see, what should I do? Maybe go to this higher magnification. There we go, so we can see its face. Smile for the camera. He's trying to figure out where to go. I think some of them have little eyes. Not all of them, but some of them have little eyes. So, yeah, I mean, they've got some sort of a nervous system. Sorry for the stupid questions. I just know very little biology. Oh, you're allowed to ask any questions you like, Bettine. And I don't judge people. And I don't think they're stupid, in any case. They're just questions. And it's not like I'm an expert on water bears, or anything other than diatoms, really, and maybe photography. I'm not even sure I'm an expert at photography. I just like doing it. I'm a paleoecologist by trade, but paleoecology means you have to know biology and also a bunch of other things like chemistry and, in my case, limnology. So I learn a lot about organisms in the process, and it's important for me to understand not just diatoms, but things that might eat diatoms and the whole environment that they live in. So this is part of that. Yeah, that's the total point of this channel. Show people stuff. Let them see things that they're not used to seeing and think about things that they're not used to thinking about. So, exactly. I don't mind questions. And, oh, what is this? Do you see that? That right there is a little inactive water bear. Maybe a molt of this guy. See how close it is in size and everything? And it's not moving. And not only is it not moving, but you can tell it's a water bear. I can see its claw. Right there, I can see... Oh, you can't see where I'm pointing anymore. One problem with using this camera. Let's see if I can zoom in on it, and then you can see the claw. See the little claw? Right there? At the top? That's definitely a water bear, and it's got the same little armor coating that this one has. So it's either the molt of something like this, or it's a dead one, maybe. Yeah, they're not bears. There's some parthenogenic lizards, I think, on some small islands, but I don't think they've been around for a real long time. So parthenogenesis for most organisms is like, it's not totally asexual. So like rotifers, all rotifers are parthenogenic, and or at least all the ones that have sexual cycles are parthenogenic. But they periodically, so it's like they're female most of the time, they go through this cycle, and then they occasionally will, a male will spawn from an egg, like they'll lay male eggs, and then they just are around long enough to have sex, and then they switch back to the asexual cycle. And copepods are the same way, where they will just have like a little loop, like a tiny little loop where they undergo sexual reproduction, because it gives them a little window where they can actually like evolve if conditions get bad or they need to basically recombine genetically. But the Bedeloid Rotifers don't even do that. They don't have any like sexual cycle in there. It's just cloning basically. And if you're wondering how the heck do they do that, how do they manage, what's kind of cool about them is that they have this really great ability to repair their DNA. And what they found was that they could bombard them with irradiation, like, nuclear irradiation, and really, really high levels or doses of it. And they could repair themselves. And not only could they repair themselves, but that even after being exposed to something that would, like way more than kill a human or most organisms at all, they would still be able to reproduce. I mean, they'd still be able to have viable offspring. So not sexually, but that's what makes them different than basically all the other organisms that are like that don't have a sexual cycle at all. Like, they don't have a little loop or a broken wheel kind of a structure to their sexual cycle, where they're just entirely asexual. And it's the only reason that they probably have managed to do that is because they are also capable of withstanding these really high radiation and repairing their DNA. And one of the things that's kind of cool that they found about this was that it's a common behavior for organisms that are adapted to these really dry lifestyle, where they're dry most of the time and then just get wet for a little bit, is that they also seem to develop a resistance to irradiation, not just UV radiation, but actual like nuclear radiation. And so water bears are also resistant to those conditions in their ton state, not in their living states. But they also are resistant to radiation as a result. And there's some bacteria that also are really resistant to radiation, and they found that they also do really well in conditions where they can dry out. So I don't know if there's some example of polyextremophiles where it's not that they intend to, it's not like they're adjusting to something that they're exposed to, because most organisms are not exposed to nuclear radiation, but they end up being adapted to, resistant to it, because they are built to withstand these really dry conditions, and for some reason that also gets paired oftentimes with nuclear irradiation. So that's kind of cool. Anyway, water bears and Bedeloid rotifers both have these environments that they live in, and similar kind of lifestyles. It's just that the rotifers don't get the kind of press that water bears get. And of course, some aspects that are really important about water bears that make them distinct. They're in their own, like way up the chain somewhere. I think they're in their own phylum. So when people say, well, what is a water bear? Is it like an insect or an arachnid? And you're like, no, it's like a water bear. They're in their own group, because they're so distinct from everything else. But also they evolved really early. And our evidence suggests that they were and have been around through all of the major extinctions. So all 5 major extinctions. And there's not a whole lot of organisms that are like that, that are alive today, that have been basically persistent through all of the major extinction events. It's like brachiopods, and I don't know if brachiopods even made it through all of them. But there's not a lot of these organisms that are like this, that basically have been around, genetically. The group's been around for a really long time. So, they don't care what we do. to the planet, they're going to be fine for a while at least, under pretty much any conditions. That sounds like some kind of anti-cancer property. It is what it is. It's exactly what it is. Am I alone? Can I handle a large raid if you can set it up? Well, Tropical Geek, I can handle whatever. I don't need any chat assistance, but I've got, I've got people here who can mod for me. I don't know what you mean by a large raid, but, I can handle people. Worst case scenario, I won't be able to answer everything everybody asks, but that's okay. So it doesn't have a chat assistant, yeah. Well, earlier Mallory was here. She checked in for a little bit, and Sarah was in for a little bit, and OpenSet's here right now. So if you want to bring some people in to see the water bears, you can. I've sort of been focused on just this little guy for a bit. I'm probably going to stay with him, especially if you're going to bring some people in, because he's big and he's easy to see. The other little guy that we saw, the transparent one, is a little bit harder to spot. But yeah, we can handle it for sure. I'm always ready. Bring him in. Large raid coming soon. Thank you, Tropical Geek. Thanks for bringing people over. I love the extra exposure, and it'll give me a chance to talk a little bit about what I do as well. Oh, somebody just came in. Paleontologizing is now hosting my stream with 67 viewers. Welcome in, raiding party. Hello, Paleontologizing. You're probably busy trying to figure out how to shut down your stream at the moment. I want to say hello to all the people who just came in. The raid, 57 people, welcome in. Right now we're looking at live stream from my microscope, and that little blob you see walking around down there in the pile of stuff is actually a water bear. And I'm trying to get him into focus for you, but he's hiding underneath a bunch of stuff right now.
Good news, everyone.
Thanks for the follow. Welcome in, everybody. Well, we can get his face right there. You go. There's the water bear. He's crawling around.
Good news, everyone.
And thank you for the follows, Paleontologizing. Thank you for the follow. You can see right there. He's got I should probably turn that off so it's not coming through twice for you guys. You can see right there he's crawling around in some lichen, and if you're wondering, oh, where did I get this sample from, I just covered that a little bit ago, but I'll try again. I went out to a local park. I grabbed some sticks off of the ground from under some trees, and there's some lichen that's actually on the sticks here, and here's some bark. And what I did was I just took a little paring knife, like this one, and I just scraped some of it off into a little jar or into a little bowl. So it's just that little bit of lichen right there. In this case, I actually took some lichen from a number of different samples. And oops, where did he go? He's up here. Sorry, he's crawling around in the back sides of this stuff, so it's kind of hard to catch him right now. Let's see, I can move out to a lower magnification. There's a little rotifer up here, slightly exposed. Right now I'm streaming from home, from my Light Microscope, and normally I look at diatoms. Today I'm looking at some water bears and rotifers and a bunch of little microorganisms that are found in these dry environments, like lichens and mosses. And here's that water bear. He's sort of poking his head around right there. He really likes to hang out on the back side of everything while I'm trying to showcase him. So we've seen some, oh, there he is down at the bottom now. We've seen some water bears. You can see it right there. Trying to get it into focus while he's crawling in 3 dimensions is kind of hard. We've seen some rotifers. We've been talking a little bit about the ability of rotifers to withstand conditions that are similar to those that water bears can handle. Really extreme dry conditions, UV radiation. They can be exposed to the vacuum of space, in their hibernating state. They're actually capable of withstanding temperatures lower than freezing, and can handle, like they've had water bears that they've put in temperatures as high as 300 degrees in their tun state, and then brought them out after about 10 minutes, even though that's a temperature that you normally make lasagna at, and they're fine. They can still reproduce and everything else. So they can't handle that in their living state, which is a misconception that people have. But they can handle it when they're in their sort of tun state. It's like a cryptobiotic state where they are temporarily suspended. So yes, I'm Diatom's Attack is my code name here. I'm a professor at Indiana State University, and I primarily study diatoms, but also other microorganisms. And normally I stream from my scanning electron microscope at the Indiana State University, and usually I have a team of people with me that help me out, because working on a microscope is kind of challenging, trying to bounce back and forth between people asking questions in the stream and you know, I gotta have my eyes in here sometimes to see what I need to see, so I can focus a little bit sometimes when it's on my screen. Let's see, I did some field work in diatomaceous earth quarry last year. It was pretty neat and very dusty. Yeah, I work in some diatomaceous earth, what we call diatomite. I have a paper out earlier this year where I described a new genus from some diatomites in Idaho, collected from some material from the Idaho Geological Survey. And a lot of the diatoms that you find in that diatomite's the same stuff you would find if you went to the hardware store and just dug around in a bag of diatomaceous earth. But the species that I found and the genus that I found were new to science. And I don't always work in the really old stuff, but my background is actually as a geologist / paleontologist, and I do paleoecology, so not dissimilar from what paleontologizing does, only I look at really small stuff. And most of the time I don't go back in time as far as paleontologizing usually does. I usually stick to maybe 10 or 12 million years and more younger than that. But, you know, today we're just looking at some living material. I like to sort of bounce back and forth. In the scanning electron microscope, we really can't look at living things very well. So I like to put live diatoms or live water bears or rotifers or things that we find in lake samples on the light microscope, where you can see them in their natural environments. There's a little rotifer stretched out over here. You can see crawling around, and we're on the lowest setting that my microscope actually has right now. But if I zoom in too much, then you won't be able to see very much of them at the same time. So, but I can. Oops, that's a little too close. Let me get that into focus, and then we'll hop in and look at them real close here. Let's see. Let's do that, and then maybe out a bit. Here we go. So here's a little rotifer crawling around in there, and over here that water bear is just persistently hiding under that thing. So the rotifers, as I mentioned, are similar. They're capable of living in similar types of conditions, and these little Bedeloid Rotifers are really kind of impressive. They can handle super high temperatures, the same sort of low pressure systems in their sort of suspended animation states. So as I mentioned, these samples are just lichen material, and we've also seen some little mites crawling around in the sample. We've seen some ciliates. There's a little ciliate. I could chase it a little bit. Oops, there it is. They're really fast. Not the kind of thing I like to usually try to keep in my field of view. It's a little tiny ciliate I'm chasing around. Oh, he's probably just outside of your range. And we've seen some little mites. And oh, here's another rotifer. and some nematode worms, which are all pretty common in these types of materials. There's another little ciliate. Zooming around. See if I can zoom out a little bit so you can see the whole field of view in case something crawls in and out of it like that again. So yeah, the thing that's really impressive about this sample is, of course, that it started off as just a piece of a stick, and then subsequently we've been able to just re-wet it, take little pieces of it, re-wet it, and then and we can scroll around in it in the light microscope and find all kinds of little organisms crawling around in it, which I think is really impressive. Normally when I present from my scanning electron microscope, I do that usually Wednesday and Saturday from 1 to 3. I don't know how long we'll stay on that same schedule, but if you're interested in seeing stuff, the light microscope has a limit in terms of its resolution of about 1,000 times magnification. And my scanning electron microscope has a resolution in terms of like magnification of something like 180,000 times. So what we're looking at here, as I mentioned, is only at about 100 times magnification. So if you really, really like to see small stuff, you should probably check out one of those streams, usually in the afternoons when I'm at work, or when I come in on the weekends to do a little bit of streaming. Those are really interesting, and if you are on a PC and you scroll down below me, you'll see in my About section there's a Discord channel there. I post some of the artwork from. I'll take those pictures from the scanning electron microscope and colorize them. Sorry, I want to come back and see what you guys are talking about. Would you use a Leica microscope, or is that not useful for diatoms? This is a Leica microscope that we're looking at right now. This is a Leica DM2500 that you're looking at. And then the camera that you can see up here on the top is actually an Olympus OMD Micro Four Thirds camera, the EM1 Mark II, which is a pretty nice camera body. And I've just got it mounted to the microscope with a little T-mount. But the diatoms are mostly analyzed on a microscope that's like this. What separates it from most of the other microscopes that you see that people are using is that it has differential interference contrast in the objective types. So not the one that we're looking at, but when I jump up to the 200 times magnification, this is actually differential interference contrast. So I can play with, tweak with the contrast a little bit, which is what's going on right there. It looks a lot better when I'm looking at a static slide because I can really get zoomed in close. I don't even have a cover slip on these. Which means that if I bump into something that's wet, it'll stick to the bottom of my lens and or my objective, and I won't be very happy about it. So mostly try to scroll around in the 10x objective because there's little chance that anything's going to reach up and touch it. Oftentimes when I'm looking at diatoms, I could just put a cover slip on it, but you can see this stuff is really three-dimensional, and if I try to put a cover slip on it, just make a lot of air bubbles. So I'm trying to avoid that. And as a result, I don't have a cover slip on it. And my optics would even be, oh, there's a little nematode. The optics would be even better, of course, if I did have a cover slip on it, because that's the way that it's designed. It's designed to have a cover slip in there. And then I would be able to jump up to a much higher resolution, higher magnification as well. So just because I, at the moment, don't have a cover slip on this sample, so I can't use that very well. You were looking for stickleback fishes. Well, I mean, that's fine. You don't, you could just save all the diatoms for me and I'll look at those. Yeah, you could always dig out some material and send me the diatoms. Have I seen any amoebas on my stick samples? Yes. So OpenSet, that's a good question. I haven't seen any living amoebas. But I do find the testate amoebas, the type that are called Arcella. There's one right there. That is the home. That right there is the home of a amoeba. The amoeba is no longer with us, I don't think. I don't think it's crawling around. Maybe it is. I don't know. I don't think so, though. Maybe that's still alive. But that's an Arcella right there. Very slow moving if it is. It does seem to be moving just a little bit, but I don't know if that's drift from vibration. No, there's a little bit of the pseudopod sticking out underneath it. Do you see that? jump up to higher magnification, that's an amoeba right there. That's Arcella. So that was a good, well-timed question. You can see its pseudopod sticking out, waving around a little bit right there. So they live in a little sort of cone-shaped testate home, and that's it. It's crawling around very slowly. It's the dark brown thing is its actual home that it lives in, or its test. It's a type of testate amoeba, and you can see a little pseudopod sticking out. So, yeah, that's Eastern time. Sorry, I'm in Indiana. We're almost all on Eastern time. So, and I do have a Redbubble site, and in Delaware, thank you for sending that. I will take a lot of the things that I see in the scanning electron microscope that are diatoms or water bears, and then I draw them. So if you're interested in getting some diatom merchandise, you can go to that Redbubble site. They make it as you order it. So there's stickers and t-shirts and mugs and all kinds of things with my artwork on it. Those are my digital drawings that I've made. And if you're thinking, oh, I'm trying to sell you some stuff so I can get rich, I will tell you that I don't take any of the money from either subscriptions or from stuff that we sell on that website. All of it comes back into my lab for student research. So despite the fact that this thing is living, it just crawls around so slowly you almost wouldn't be able to tell if I wasn't just sitting on it. But you can definitely see the pseudopods underneath it kind of repositioning themselves a little, right? You see the little pieces of it that basically, like a snail, it kind of just crawling around in its little home. You know, the slides aren't moving, and that stuff you see in the background that's wiggling around is actually bacteria. So, Sid Hersey, of course. Yeah, OpenSet's here as my mod sometimes. He just comes out and hangs around, you know, tries to keep the stupid robot spams from making me famous, because I guess I'm not famous enough, but yeah. So it was a good question, though. I probably wouldn't stop to look at this thing if you hadn't asked it. I keep seeing their the little testate amoeba homes, the Arcella homes, but I never really stopped, slow down enough to actually watch and see if they're crawling around. That one, you can see its little pseudopod coming out in the back right there. It looks like bubbles or something oozing out. That's actually just its little pseudopod legs, like amoeba legs crawling around. And it's got a stack of junk on the back side over there, on the left-hand side. Came in on Paleontology's raid. Thank you for coming in. And also thank you everybody for all those follows. I can't keep up with it while I'm trying to look in the microscope at the same time. So, but I really appreciate it. And I want to thank also Tropical Geek for bringing the raid over to our channel. I'm not usually on at midnight on a weekday, but sometimes I just get on my microscope late at night to get a little time, you know, a little time, quiet time, away from the craziness of my day, and check out things on the Light Microscope. So I'm really glad you all came over to visit. I know, you know, Paleontologizing does great stuff over there, looking at dinosaurs and paleontology. I do sort of like paleontology. I mean, that is sort of my field, although I think of myself more as a paleoecologist than a paleontologist, because I usually try to just apply what I learned from the ecology of the organisms through their fossils to reconstruct things like drought or evolution patterns. So, but you know, big stuff, little stuff, it's just a matter of scale. A lot of it's just as interesting. We were talking about how the water bears have lived through all of the 5 major extinctions. So we were just talking about that before you guys came in. Self-care is taking a trip through the microcosmos. Yeah, Andor, that's true. For me, it is. You know, a lot of times when I'm working on the microscope, people are like, oh, how can you stand to spend all this time looking in the microscope? And I'm like, hey, you don't understand. This is like a little Zen world, you know? Like, you can get away from everything and you can just spend some time looking at this sort of microscopic landscape where there's all these alien beasties. Look at that. I found a little claw, an insect claw. Just hanging out. I probably accidentally scraped its body apart with my knife when I scraped this pile of stuff into the bowl when I started. But we switched to this sample when the other one started to get a little dry, and I hadn't even looked through it yet. And we already found a couple of water bears just kind of scrolling around through it. There's another rotifer right there. It's a Bedeloid Rotifer. As I mentioned, they're kind of like the water bears with respect to their durability, their ability to handle all kinds of crazy conditions. And I think there's something right there all rolled up. That might be a water bear or it might be a rotifer. I'm not very good at identifying them when they're in their little ton states or they're in their little rolled up states. It kind of makes me think it's a water bear. You see that little thing sticking out on the top up there that looks like a claw? I'm not sure, but that is basically how they look when they're in their little rolled up states. They just look like that. They're not super interesting. They're like a little ball of junk with some claws sticking out occasionally. So, oops, there's a water bear. Didn't take long to find that one either. You can tell it's a water bear pretty quickly by their movement. So if you can see it very clearly, their front legs are always just like groping for something. You know, they're exploring their landscape with their front two claws, and if they get a good grasp on their back, so they have 8 legs, if they have their back 2 legs attached to something pretty well, they usually have all 3 of their legs, all 3 sets of their legs kind of like groping around for resources. So you can see that right there pretty well. I think you can get a good look at it, and then I'll see if I can zoom in a bit more. Yeah. There you go, a nice clean shot of just the water bear. This one's not hiding quite as much underneath the pile of junk yet. There's its back legs. They're kind of like, sort of like a tail almost, but they do have distinct claws on the ends of their feet. So they aren't quite like a tail, but they are kind of welded together a bit like a shrimp tail, kind of, when you look at them. The back 2 feet. They don't have full articulation the way that the other legs do. So they always are changing their elevation, which makes them really challenging to keep in focus. So especially when I get zoomed in and the depth of field shrinks, depth of focus shrinks. What age strata do I usually work in? Miocene? Sorry, a little slow answering your question. I'm usually in the Holocene, but when I look at diatomites, I'm usually in the Miocene. Late Miocene, Pliocene, Pleistocene. So I work a lot in East Africa, looking at diatoms and trying to use them to help reconstruct environments where humans or hominids lived. That's been the bulk of my research in the last decade or so. And I work in the Rocky Mountains, the Sierra Nevadas. I work in Mount Everest area. I've worked in the Pantanal in Brazil, and I work in a whole bunch of sites in the East African Rift. And I'm not limited to those sites. Those are just the places where I do most of my work. I have new research that's sort of developing in Guatemala, and I just follow wherever people need diatom work done. And then again, the Miocene stuff that I've looked at has either been in Africa or in the western part of the US, in Idaho basically, or in the Lake Idaho Formation stuff. Yeah. So diatoms have a record that's basically as long as dinosaurs. It goes back basically through the Jurassic. into the Jurassic at least. But most of that stuff is marine. I don't think they get into the freshwater systems, or at least they're not abundant in freshwater systems until the Eocene. So, and then they really start to take off basically in the Miocene, and diversify and get kind of interesting. So, that's basically why I'm looking at them there, and that's where the diatomite collections usually are the largest, sort of Miocene, Pliocene, Pleistocene ranges. So, but a lot of my work actually takes place in the Holocene. I look at stuff that's lake sediments, so, before they're diatomites, they're just lake sediments, and those things are, 10,000, 12,000 years old at the most. The majority of all oil deposits in California are actually from diatoms. Yeah, I would believe it. And I think probably because it's such a porous rock, probably a lot of the oil deposits are stored in them as reservoirs as well. It's kind of a neat scenario, because they do make the sapropels, the oils that ultimately become oil, when they get transformed through burial. But they have some people in East Africa that are sort of exploring the diatomite, the sort of modern diatomite deposits, or they're trying to dig down into Lake Tanganyika, for example, for oil, or Malawi, and they can get some out of there. I don't know that the deposits would be extremely valuable if it weren't for the fact that they're trying to produce them within their own countries. It's not like, we can get off of the Gulf Coast or something like that, but, I don't know how productive those reserves are, but yeah. This little rotifer's trying to creep in on our story over here. So this is I actually collected about 4 or 5 different little sticks, and I shaved a bunch of lichen off of all of them. So this isn't just like purely one stick, but I've had a lot of luck with sticks. I'm just gonna put it that way. I spent a lot of time looking around for water bears in a bunch of different environments, and because they're called moss piglets, I thought, oh, they'll probably be in mosses. And I don't know how many lot mosses I looked through. I never found any. I don't know if they were just like too carefully stuck in the mosses. It's kind of hard to get the material out of the moss. But it was a real challenge, because I was thinking, oh, they'll be all over these mosses. And I kept trying all these different mosses and never found any. And it wasn't until I switched to just looking at the lichen and sticks, and I just did it out of frustration. I found a stick in my driveway, and I was like, I'll give it a shot. And there they were. He's hiding in something? Yeah, they're always hiding in something. Yeah, I took a stick out of my driveway that had a bunch of lichen on it, and I thought, I'll just give this a try. I scraped some of it off. The nice part is I could bring it into my house, and I could just kind of scrape at it. Hey, Mama Bonbon. I could just scrape at it a bit until I got the pile of lichen. So if you're wondering what it looks like a bit like this. It's sort of like a lichen stew. It's kind of hard for me to show because my camera's mounted on top of my monitor right now. But it's just like a whole bunch of junk at the bottom of a little bowl, and it's kind of green colored from the lichen and the algae combo. And I just pipette a little bit up in, and basically can drop that right onto a sample, onto a slide. And there's usually a couple of water bears, and then a bunch of rotifers and some nematodes, and whatever else in there. Hey Lex, I know that there's supposed to be water bears living in like aquatic systems, but I've never seen any in lake sediments, and I've never seen any in lakes themselves. That I don't look at the mosses in lake systems. That's probably why, because they're usually too shallow for what I want to explore. But but they do also live in ocean systems. So, but I don't work in the ocean very frequently, or if all at all, really. Just occasionally I take on some projects that are ocean related, but I haven't actually taken any samples from diatoms from the ocean directly. It's usually people sending them to me. I work a little bit with Pacific Plankton. She sends me some materials now and then from San Francisco Bay, and I should send I don't know if we send a shout out to the microscope squad, including OpenSet, who everybody probably already knows, but these are my little squad of microscope streamers. Usually more coastal. Radiolarians and forms. Yes, yeah. They would have to be living in basically shallow water. I don't think they can swim. They've got the little claws so they can attach to some substrates, and so they would be somewhere along the coast, probably living in lagoons more than in the open oceans. But you should be able to find them if you went around on a beach environment somewhere. You catch the right settings for them. I just don't know what those settings are. Also, I live in Indiana, so. Oh, diatoms? No, diatoms are everywhere in the ocean. The reason that they're limited to the coast more frequently is because they have a silica as a nutrient resource requirement, and silica in the oceans is quite low in terms of concentrations, except for the Southern Ocean, which gets a lot of nutrient upwelling and also a lot of silica from Antarctica. And so if you were to go out into pelagic environments in the open ocean, you would if you went into the ocean gyres, you probably wouldn't find any diatoms, because nutrients are low, and especially silica and phosphorus are low in those settings. But if you were to go into the Southern Ocean, like even in the super deep water out there, you'd find diatoms everywhere. They're the dominant organism in the Southern Ocean and in parts of the South Atlantic, and also along the coasts, and probably in the Arctic. They can live frozen into sea ice, and the diatoms live in basically all these crazy environments as well. There are extremophiles, just underappreciated extremophiles, because the single organisms usually can't live in a wide range of environments, but as a group they live in basically all the same crazy environments as these guys. You can find diatoms living in mud puddles, that form in the street when it rains. You can find diatoms living in tree bark, in soil. Anywhere there's basically sunlight and a little bit of nutrients, you'll get some diatoms living. But they can also live frozen into the sea ice in these hyper cold, hyper saline environments. They find them in Antarctica, in the coldest, most high UV radiation exposed parts on the surface of the Earth. That they're wherever there's water and some nutrients, you'll find diatoms living. If there's no nutrients, though, they usually get outcompeted by other types of organisms. So they do well in coastal environments, particularly because the nutrients are available to them. But they're not depth limited. So, there are diatoms that are depth limited, the benthic diatoms. But there's pelagic diatoms that live in the deep oceans. They just, they need to be where there's nutrients. So, coccoliths are usually the things that you find in low nutrient environments more so than anything else. Forams can be in those environments in the warmer water usually, but diatoms and forams are found together in a lot of settings. So yeah, they're also extremophiles of a type. If diatoms are underappreciated extremophiles, which are the overappreciated ones? Yeah, water bears probably. They get all the press, It's like trees get all the press for generating oxygen, and diatoms generate more oxygen than trees do. And water bears get all the attention for being extremophiles. I think the legitimately underappreciated extremophiles are probably the bacteria and the archaea. Nobody cares about them because they're not charismatic. But water bears are very charismatic organisms. Diatoms are just sort of underappreciated because people don't know what they are capable of, what kind of environments that they can live in, and they tend to think of them as just being food. Like they're kind of like the grass of the ocean, rivers, and lakes. So people think of them more like they're just a food source. But there's a lot of really interesting diatoms and interesting environments that they can live in. So really some really extreme conditions for algae. They live on cave walls, subways, and caves where there's just light from the artificial light. They find diatoms growing in those settings. There's a little rotifer all dried up. And also, you probably heard that water bears can go like, 80 or 100 years being dried up with no food and come back to life. Well, I'm here to tell you they've taken diatoms out of herbarium collections where they were pressed into books, and they'd been there for 150 years, and they put them into some nutrient-rich water, and the diatoms came right back to life. So, people just aren't aware of that setting. But diatoms have also been pulled out of the clouds. Living diatoms have been pulled out of the clouds, being blown around as dust particles because they're dust-sized particles, and they colonize water bodies that way. So it actually gives them sort of a, you think about, they're smaller than water bears. They can get blown up into the upper atmosphere, and they can encysst. And then when they come out of that encysted state, they can fall into a water body, and if the conditions are good for them, they can come right back to life. Yeah, Skyatoms. I get really excited by that story when they put these sort of like butterfly nets that they towed behind, like plankton nets that they tow behind planes, and pulled them through the clouds to try to see what's in there that's particulate matter. And they actually found a lot of different algae type that get carried up into the air, and they're living cells, not just diatoms, but lots of algae. And I think that actually makes them really impressive. I often tell my students this story, that you could take a pot of water and put some nutrients in it and stick it on the roof of your house, and if you come back in 2 months, it'll be full of diatoms, it'll be full of algae, and you have to wonder, how does that stuff get there? I mean, it was pure water when you started. It could be raw pure water out of my special raw pure tap here at home, and a bowl that was sterilized. And there's definitely no diatoms in it when you start. Put some nutrients in it, and they'll be growing in there by the time you're done. So, but that's kind of cool. It tells you that that's how, it's a mechanism for them finding and sort of colonizing these environments and being distributed, into environments. There's some challenges to that, of course, which is that there are diatoms that are basically endemic, but or at least we think they are. But I think that is pretty good evidence for them being extremophiles. I have some rocks over here that I pulled out of a river 2 or 3 weekends ago. Yeah, a month ago, actually. They've just been sitting in a bag on this sort of table next to me. And I can guarantee you that if I wet that thing up, the rocks up, and scrape them a little bit, the diatoms on there would come right back to life. So, they can definitely handle being dried out. They find them in desert soils. They find them in, like all kinds of crazy super acid conditions. There's an acid mine drainage site around here that gets pH 1 in the water that comes out of it. Diatoms are totally living in it with no problem. So, I mean, pH 1's pretty crazy. I think that water sometimes gets down to 0.5 or even close to pH of 0. So, I've ever thought of digital light microscopes that have multiple lenses doing 3D images. A confocal microscope, you mean? I don't have a confocal microscope. The biology department upstairs from us at school has one, but I don't actually have a confocal. I have an SEM, so I don't need one of those for many things, but it might be really neat. Maybe over break I could figure out if someone. So one of my friends here that shows up, Sarah Losaurus, is a lizard expert, and she works in the biology. She's a grad student in the biology department above us. Maybe she can get me into the confocal microscope that they have, and I could put some water bears on that, and maybe we could stream from it. We'll see. Might be fun to try. I've never used a confocal microscope. I don't really understand why I would need it if I had a scanning electron microscope, but I guess they would be really useful if I needed to look at something living, like a water bear or a rotifer. It might be it might be a better way to look at some of those things than the SEM, because in the SEM, they're in a vacuum. And I've got to coat them with gold, and a lot of the things like the rotifers and the water bears, when they dry out, they just look like little trash bags with some parts sticking out of them. So they're not very charismatic when they're dead, and they've been coated with gold and they've been stuck in a vacuum. They're definitely not the cute little things you see crawling around or spinning their cilias in my samples. So, but it would be fun to try. I don't have access to one. For diatom work, we really don't need a confocal for most. I can't think of very many cases where I would need a confocal microscope, but that doesn't mean I don't want to play with one, that I don't want to see what they're like. They sound like they would be really neat to give a shot to. And then we could make some 3D images out of that probably. I'll see if Sarah has any access to that or not. Alright, well, I wasn't planning on streaming all night. Usually I like to end my evening streams by raiding one of my friends. Oh, there's a water bear. He just came in right at the end here. Nice. Wasn't looking for him, I just showed up. Confocal is the most fancy microscope I've been able to play with. Yeah? Well, you know, when the pandemic is over, if you want to come to Indiana and play with my scanning electron microscope, you can. You know, I'd definitely let you check it out. We do a little co-stream together or something. That would be fun. You know, a geologist who's doing art who I would like. Oh, is it Ashurna? Is she on? Is that who you're talking about? Because we do need to find someone to raid. And I haven't looked at the list yet for people that I'm friends with. Yeah, Ashurna's on. Actually, I haven't had a chance to raid Enceladiosaurus, and since we got some people coming over from paleontologizing, maybe that would be a good person to raid instead. I've raided Ashurna a number of times. We're friends. I mean, I think she does a great glass blowing stuff. Let's see. Can we see what? Let's see what Enceladiosaurus is doing. She's in science and technology right now. Let's see what she's got going on, and then maybe if she's doing something cool, maybe we'll come over there. Sometimes she's just doing games, but I think she's actually answering questions and stuff. Oh no, she's hosting somebody else, so she must be at the end of her stream. Oh, she's interviewing a game developer. Confocal beam scanning microscope. Yeah, I think that's, they're not the same thing. A confocal microscope is, doesn't take place in a vacuum at all, I don't think. A scanning electron microscope scans with an electron instead of light, so it doesn't need to have confocal approach. Maybe confocal microscopes do have some sort of a scanning opponent. Let's see. Oh, you're looking at a Wikipedia page. Okay. Well, like I said, I've never used one, so let's raid Ashurna then. We'll do that. That sounds like a good deal. William Bradlow Amos and John Graham White and colleagues are working at the Laboratory of Molecular Biology in Cambridge. Oh, I see. It's in development in 1985. Yeah. Okay. Well, let's go ahead and raid. Seems how we're getting to that point. And I want to thank everybody for coming in with paleontologizing, and also the people who were here hanging out with us before. Especially want to thank Shirna. Yeah. Especially want to thank OpenSet for hanging out and handling questions with the raid, and Tropical Geek for suggesting people come over here. All of my regular viewers who are on, and all of the followers that we've picked up tonight. Space Nerd, Little Bungorv, Cloudbone, Uppity Mallard, Strange O Mango, Feedy, Jetpack Boy, Charlie's Dragon, Paleontologizing, of course, Sid Heresy, Blue Spider Monkey, Barbarian Hamster, E Studio, Fetal Femme, Matt M33, and then two people who came just, Matt M and Granite Crow, who followed me just before the stream started. So check us out again. I'll be on Saturday with a live stream from the Scanning Electron Microscope. I think I'm actually going to start the stream on Saturday at noon, and I'll have my lab assistant Mallory with me. We'll be looking at some, I don't know what we'll be looking at, some crazy stuff probably in the Scanning Electron Microscope, and then we're going to raid somebody around 2 p.m. for part of the Artist Back Alley. So friends Little Chook and Miss Daisy D, who are some of the primary people behind the Artist Back Alley, and I work with them. I'm a part of the Artist Back Alley, and I wanted to be engaged in their raid train that they're doing on the weekends. So I moved my schedule around a little bit for Saturday so that we could stream from noon until 2, so I could end my stream with a raid that goes right into the next person in that raid train. So yep, Andalore, thank you for hanging out again, for hanging out with our night stream here from my home microscope, and we'll catch you guys all again. I'll try to do a little bit more night streaming now. Your appreciation for diatoms has gone up 1,000%. That's a lot of percents, paleontologizing. Thank you a lot for bringing your people over. Hopefully we took good care of them while they were here, and we'll catch you guys later. Please check out any of my sort of squad of microscopic streamers if you're interested. OpenSet should be doing a little bit more streaming recently, and my friend Pacific Plankton, it's her birthday tonight. She usually streams tonight, but she can't because it's her birthday. So she's at home having a good time. But you should check out both of their streams, and we'll catch you guys later. We're going to go raid. Thank you.